standards for the beneficial use and disposal of sewage sludge regulate the quality of sewage sludge. Under the Clean Water Act, EPA set pollutant limits that must be met when the sludge is land applied or disposed of. Compliance with these pollutant levels is evaluated by sampling and analyzing sewage sludge before it is land applied, incinerated, or placed in a surface disposal site. Proper sludge sampling techniques are the topic of this video. Throughout these demonstrations, we will focus on two important sludge sampling challenges: identifying the correct sample location and collecting a representative sample. Because there is a wide variety of sludge processing methods, you may be sampling a liquid or solid material from several types of sampling locations, including pipes, conveyor belts, and piles. The most appropriate sampling location is the last point prior to the sludge leaving the treatment plant. This is where the sample is representative of the sludge which is hauled off for final use or disposal. Consider safety and accessibility in identifying the sampling location. Before anyone collects an environmental sample, whether you are a technician or inspector, the first thing you need to do is your homework. Sampling and analysis is very expensive, and if you don't get the right sample, you've wasted your time and your money. How you prepare for sampling will vary depending on your objectives, but there are some preparatory steps that everyone needs to take. One useful tool to make your sampling event most efficient is a sampling plan. Here are some key issues you need to consider before you go. Determine which parameters you are collecting samples for. At the federal level, the regulated pollutants vary depending on the final use or disposal of the sludge. Identify the types of sludge treatment units at the facility so that you have an idea of where to collect samples and the equipment you will need to collect a representative sample. Identify the sample collection techniques, either grab or composite, needed to get a statistically representative sample. The amount of sample required depends on the solids content of the sludge and the detection limit of the analytical technique. Check with your lab and make sure you collect more than enough sludge for analysis. Typically, sludge sample volumes range from 8 ounces to a quart or 250 milliliters to 1 liter. The appropriate sampling equipment depends on the type of sludge being sampled and which pollutants will be analyzed. Think about possible sampling errors and how you will avoid them through quality control procedures such as equipment rinsate blanks and trip blanks. Proper quality assurance usually requires collection of extra samples. Prepare for safety issues. Make sure you have standard personal safety equipment available, such as steel-toed boots, hard hats, safety glasses, and gloves. If you are a compliance inspector, you will also study applicable permits and fact sheets, compliance reports, previous inspection reports, and past monitoring data. After you've done your research, you're ready to schedule your sampling event. First, alert the lab. Let them know what analyses they will need to run and when you will need the results. Also, confirm sample volumes and quality control procedures with the lab. Make sure you plan your sampling trip so that you will get the samples to the lab on time. Remember, pathogens only have a 6-hour holding time. You will want to contact the facility to make sure the sludge is ready for disposal. Next, gather all the sampling equipment and sample containers that you might need. In general, your equipment will include stainless steel buckets, trowels, scoops, and pitchers. You may also need an auger for collecting samples of sludge from bins, compost piles, drying beds, and stockpiles. Stainless steel is generally acceptable for any sampling. If you're concerned about metal contamination, then use plastic equipment. Remember, plastic equipment may not be used to collect samples for organic pollutant analysis. Sample containers are either plastic or glass. If you're collecting samples for metals analysis, you can use either, though plastic is preferable because it does not break. When collecting samples for organic pollutants, Glass containers with Teflon lids are used because most plastic containers have the potential to contaminate the sample with organics. All equipment and containers must be clean. To wash sampling equipment, do the following. Wash with laboratory-grade detergent and water. Rinse with tap water. Rinse with a 10% acid wash. Rinse with deionized water. Rinse with an organic solvent like hexane if you do not have time to allow the equipment to air dry, and cover the equipment with foil to prevent contamination. If you are sampling for pathogens, you need to use sterile sampling equipment and containers. You also need coolers and ice, bubble wrap to protect any glass jars, labels, plastic bags, tape, markers, chain of custody forms, and seals. Ice is very important since it preserves the samples in the field. Samples must be chilled to 4 degrees centigrade until they are analyzed. Finally, you are ready to go collect samples. Let's start by taking a sample from a digester. Digesters generate a liquid sludge that typically ranges from 0.5 to 4% solids. This plant has an anaerobic digester, which discharges liquid sludge to tanker trucks that haul the sludge to land application sites. The key is to find a safe location to collect a sample before it is loaded into the truck. The truck loading port would be good, except that the pressure under which the sludge is pumped into the trucks is too great to allow us to safely collect samples. Most facilities have a sample tap inside that they use. You must make sure that you sample from the line that's discharging the final digested sludge and not raw or recycled sludge or supernatant. The discharge line and sample tap must be purged of old sludge before samples are collected. Make sure to let the sludge flow until the volume of the pipe has been replaced with fresh sludge, or the discharge sludge temperature matches the digester temperature. This ensures that the sample collected is representative of the final sludge that is leaving the plant. The first sample collected is for pathogens using a sterile sample bag. To collect a single grab sample, place the sample container directly under the sludge stream. Nothing but the sample itself should touch the inside of the bag to prevent contamination. Leave a half-inch of headspace at the top of the container to provide room for expansion in the event of gas production due to biological activity, unless you are collecting a sample for volatile organic pollutants. Liquid sludge, especially anaerobically digested sludge, is particularly susceptible to gas production. For volatile organics, you want to prevent the pollutants from volatilizing in the container. In this case, fill the container to overflowing. Put the cap on and flip the bottle to make sure there are no air bubbles. To take a composite sample for metals analysis from a digester, collect a series of equal volume grab samples called aliquots periodically throughout the entire time the sludge is being discharged. For example, if the sludge is discharged over a 6-hour period, take 6 aliquots at 1-hour intervals. Pour each portion or aliquot into a clean bucket. The bucket must be kept on ice. When you are finished, thoroughly mix the sludge in the bucket and quickly fill the sample container. If the sludge solids are settling rapidly, which is common with raw primary sludge, vigorously mix the composite sludge and remove sample aliquots with a ladle. Pour the entire ladleful into the container. Next, we will demonstrate sample collection of dewatered sludge being discharged onto a conveyor belt. Conveyors are commonly a convenient location to sample sludge prior to final use or disposal. Be careful when collecting samples from the moving conveyor belt. Safety should always be the first concern during a sampling visit. At this location, 3 types of samples will be taken: pathogens, volatile organics, and metals. We will collect the samples for pathogens and volatile organics first. For these parameters, it is important to collect the sample directly into the final container without transferring it. Again, pathogen samples must be collected in sterile containers with sterile equipment. Use a sterile scoop to collect sludge off the conveyor and pack it directly into the sterile sample container. Volatile organic samples must be collected to minimize volatilization of the pollutants. Fill it completely, packing the sludge in and filling it to overflowing so that when you put the lid on, there is no air space for volatiles to escape. Remember, samples for organic pollutants require glass containers with Teflon-lined caps. To collect a composite sample from a conveyor, take a series of aliquots over the entire discharge period and mix them in a bin or bucket. Keep the samples on ice until you're ready to fill the containers. Mix the sludge thoroughly, stirring and folding for several minutes. Next, fill the sample containers. Biological activity causing gas formation is less likely in dewatered or dried sludge, so leaving room for gas expansion within the sample container is less critical. Next, we will demonstrate how to collect a composite sample from a bin. The same sampling procedures can be used for a sludge drying bed, a truck bed, or other similar container. In your logbook, diagram the bin, dividing it into 4 quadrants, and mark the sample collection points. In order to collect a representative sample, equal sample volumes should be collected from the center of each of the 4 quadrants in the bin, using a scoop if the bin or drying bed is shallow, or a coring device that allows you to collect a sample the full depth of the sludge. These samples should be combined, forming a sample that is representative of the entire contents of the bin. Using a coring device, take a full core from the center of each quadrant in the bin. To use the coring device correctly, position the coring device, press and twist down into the sludge, twist and remove. Repeat until you reach the bottom of the bin. If you are sampling from a drying bed, take care not to get too much sand in the sample. Again, for a composite sample, the aliquots need to be mixed thoroughly to ensure that the sample is representative of the entire bin of sludge. When the sample is too large to mix in a bucket, you can mix the sludge on a plastic sheet. There are a variety of mixing techniques. This one involves layering the sludge, folding it onto itself, and mixing. This 2-step process is repeated 5 or 6 times before filling the sample containers. We will demonstrate another mixing technique for large volume samples later. Now, we're going to demonstrate sample collection procedures from sludge piles. First, a compost pile, and then a stockpile. Again, because we are collecting samples of the final sludge product before it is land applied, samples must be collected from piles which are fully cured and ready to be hauled off-site. For a composite sample, full cores of sludge should be collected from randomly selected sites distributed along the length of the pile. No less than 4 cores should be collected and mixed together. Again, you should diagram the pile and note the sample collection points before you start. For pathogen analysis from a compost pile, use sterile gloves or a sterile scoop. Dig deep into the pile. Take a grab and put it directly into the sterile sample container. A stockpile may be sampled using the same general procedures as the compost pile. It is usually more challenging to collect a representative sample from a stockpile because it usually is comprised of sludge that has been piled up over a longer period of time. Its shape is less uniform, so gauging how many sample aliquots to take from each section so that they are proportional to the mass of the section is difficult. The size of the pile can make it difficult to get aliquots from all depths. You can also sample the final sludge product on a front-end loader before the sludge is dumped into a truck that is going to haul it off-site. The cone and quarter method is another procedure commonly used to reduce a large volume of sludge to the volume needed for sample analysis. Pour sample aliquots onto the center of a clean plastic sheet, forming a cone-shaped pile. The pile is then quartered, and 2 quarters are randomly selected to be discarded. The remaining quarters are reshaped into a cone and quartered. This procedure is repeated until the sample volume is roughly the amount needed to fill the sample containers. Another sludge compositing technique involves taking subsamples. After collecting the sample aliquots, mix them all together on a clean tarp, breaking any large clumps. Using a flat-bottom scoop or spatula, the cone-shaped pile is divided randomly into smaller piles. Then, one of the smaller piles is used to fill the sample container. It is very important to use a flat-bottom instrument because a curved bottom instrument will favor particular particle sizes, biasing the final sample. Before leaving the facility, each sealed and labeled container should be prepared for transport. Wrap the lid a few times with tape and affix the tamper-proof seal. Wrap glass containers in bubble wrap to protect them from breaking. You may also want to put them in lock-seal bags if you don't use waterproof ink. Then, put the samples on ice in the cooler. In your logbook, make sure you record the facility name and location, the date, arrival time, and the name of the facility contact. For each sample, record sample ID number, the date and time, sampler name, sample location, parameters being sampled for, volume of sludge collected, sample preservation, and any interesting observations about the site or condition of the sludge. Before sealing the cooler, make sure the chain of custody form has been properly completed. Once the samples have been sealed and packed, all that's left to do is ship or deliver the samples to the lab and clean the equipment one last time. This video has demonstrated some of the more common techniques used to collect samples of sewage sludge. Some key points to remember are: sample from a location that will best represent pollutant concentrations and pathogen levels in the final sludge product. Plan your sampling trip thoroughly so that your samples produce valid results. Know the facility you're sampling and the sample techniques you need to be prepared to do. Contact your lab. Make sure you bring the right equipment and enough sample containers, and follow quality control procedures. Make safety your top priority.